friendly version of the title is Unraveling Hairballs. When I explain to people what I do, this sort of makes sense to people. And the basic idea is I'm using protein modification data to look at signaling pathways. This study is a collaboration between Cell Signaling Technology, Mount Sinai School of Medicine, Avi Mayan's group, and some colleagues at the University of Montana. Importantly, we're funded by the LINCS Consortium, and I appreciate the opportunity to speak here, so I must thank the organizers as well. The goal is to take mass spectrometry data, which is full of holes, meaning mass spectrometers grab a peptide for analysis, then they skip a few, and then grab another one. So if you're comparing an entire data set, there's a lot of missing data. So we have to deal with that. And the way we do that is we use penalized, pairwise, complete statistical relationships and this machine learning algorithm called TSNE. And then we use the clusters derived from this to filter protein-protein interaction edges, and we call that a cluster filtered network. The idea is that post-translational modifications indicate signaling pathways, and so the correlations between the modifications, the co-clustering between the modifications, tells us something about signal transduction. So here's what I'm going to tell you. We have a study that integrates analysis of different modifications, phosphorylation, methylation, and acetylation. And we do rigorous clustering, and we use cluster-filtered networks and then shortest path algorithms to elucidate specific signaling pathways. The network property betweenness identifies hubs that we think are nodes for signal integration. And we find that phosphorylation and acetylation seem to play a dueling or antagonistic role for cytoplasmic signaling proteins, which I didn't expect. We know that histone modifications are, there's different histone modifications, but I didn't know this for cytoplasmic proteins. Finally, we have insight into the way this drug geldenomycin works. It's a HSP90 inhibitor, and it seems to affect this dueling post-translational modification problem. The data were acquired at Cell Signaling Technology by Clarissa Rykova. And which button do you push for the pointer? Oh, yeah, here we go. So it's Tandem mass tag experiment. So because mass spectrometers sample peptides stochastically, when you want to do a treatment to control experiment, it's difficult because you have a lot of missing values, so the data don't mesh. The way this works is you have different samples, six plexes in this experiment, now up to 10 or 12. You digest the peptides, then you covalently modify the peptides with a mass tag. Then you mix the samples, so when the detector takes a peak, it'll take the mixture, and then on MS2 or MS3 fragmentation, the mass tag can be resolved, and you get a true treatment to control ratio. We have multiple experiments, however, so we still have missing data in the whole data set because each experiment has the stochastic sampling problem. In addition, we have modification-specific antibodies for tyrosine phosphorylation, serine threonine phosphorylation, arginine, and lysine methylation, and lysine acetylation. The data, this is a quick draw thing here. The data is lung cancer cell lines. So small cell and non-small cell lung carcinoma compared to normal tissue. And again, we're looking at ratios here. And then a drug treatment study where we have 6 cell lines, cancer cell lines, and 4 drugs, 3 NIBs, which are kinase inhibitors, and geldenomycin, which is an HSP90 inhibitor. And these are time course experiments. The data look like this. This is a time course experiment, and you can see IRESA inhibits the phosphorylated EGFR, which is blue on this heat map. So up is yellow and down is blue in these experiments. So the hypothesis, this thing is irritating me here. The hypothesis is that clustering of protein modifications under different conditions, reveals patterns specific to the system under study, which in this case is lung cancer cell lines. And we can use these clusters of post-translational modifications, and if clusters, and so we hypothesize that PTM clusters that contain proteins known to interact identify functional signaling pathways, because signaling pathways involve post-translational modifications. Treating the missing data as missing works. Imputation doesn't work, calling them zeros doesn't work. So combined data are almost 80% missing. So we calculate statistical relationships using pairwise complete observations, penalizing the incomplete observations in dissimilarity or distance matrices. And then we use T-distributed stochastic neighbor embedding, which is a machine learning technique derived from the field of pattern recognition, to identify clusters as nearest neighbors in the embeddings. And we also learned about, from Neil Clark, penalized matrix decomposition, another fancy matrix factorization approach, and then another TSNE step for large clusters. A three-dimensional version of the embedding of these data looks like this, with different clusters painted different colors. And the data, this approach has been evaluated in a couple of publications, and Lawrence van der Matten, who is the writer of the TSNE algorithm, helped with the early publications. We then modeled the data as networks. Here is a version that the proteins are shown in black, and the modifications decorate them. And you can see the correlation edges. Yellow is positive correlation, blue is negative, between the modifications. So this is what it looks like, and we've used R to do the calculations, and we talk to Cytoscape through R Cytoscape. So we call a co-cluster correlation network modification sites that clusters together. They have to co-cluster using TSNE, and they have to pass a threshold of a Spearman correlation of some value. And it turns out between 0.5 and 0.67 works really well. So here's the entire co-cluster correlation. It's a bit unwieldy to graph and to study, but we have it all in R, and so we can use that. And so what are we going to use it for? We're going to combine this information with protein-protein interaction networks. So here's how a cluster filtered network works. We have protein interaction edges from a variety of sources. We're focusing on physical and pathway interactions, not text mining, because we want to look at signal transduction pathways. We found that excluding genetic interactions, which could be indirect, is actually more informative. And then here's the important bit. We filter the protein interaction edges to show only interactions between proteins whose modifications co-clustered. So here's a little diagram to show you what I mean by that. We have the hairball. So this is not a hairball, but obviously the PPI networks are hairballs. And we have modification co-cluster correlation edges. And so we filter out interactions that don't have modifications that co-cluster. That's what we call a cluster filtered network. Here's an example here of the tyrosine kinases in the data set. This is the hairball. Tyrosine kinases are well studied, and there's a lot of edges. And when we filter the edges, say those that have modifications that co-cluster, we get far fewer edges. So it's a way of untangling the hairball, basically. So what I'm going to tell you next is focus first on the drug treatments. And so we see links between kinase pathways and other post-translational modifications. And we're going to focus on the post-translational modifications that changed after drug treatments. And so in this network, I've graphed nodes that change most as bigger, and the color indicates the direction of the change. So blue is down and yellow is up. Crisatinib targets MET and ALK, and these are big in blue because they're the targets. Their modifications go down. And what falls out of the data, this is just what falls out of the data. These are all the targets. So these are all the shortest paths between the targets and all the proteins whose modifications changed significantly in the data after treatment with crisatinib. MET and ALK, especially MET, links to cytoskeletal proteins, so cadherin, delta-catenin, cortactin, RAC, actin. And so that's kind of interesting. MET is often associated with metastasis and migration and adhesion. The EGFR is not thought to be a target directly for crisatinib, but it is also affected, and the data also suggest how a path leading to crosstalk between these two receptor tyrosine kinases might work. So we go from IRS1 to GAB2, another adapter protein, to the Sark family kinase lin, and that could affect the EGFR part of the pathway. So back to IRESA, which targets EGFR. EGFR is big in blue, so it's inhibited a lot. As I showed you before, the phosphorylations go down. A number of downstream targets go down, and these are a number of kinases and phosphorylation sites. What falls out of the data is a number of yellow nodes indicate acetylation. So the acetylation is going up while the phosphorylation is going down, and we have these acetyltransferases as part of this co-cluster, cluster-filtered network. We have a set of proteins that are affected by modifying these two modifications. So their acetylation goes up, indicating by increasing yellow in this time course of IRESA, and their phosphorylation, the same protein is phosphorylated and acetylated, phosphorylation goes down at the same time. And this, again, this composite path, shortest path between affected proteins indicates some approximation of the signaling pathways that might be active during this time course. This then informs us a little bit about the cryptic drug geldenomycin. So it's an HSP90 inhibitor, and this again is the composite shortest path from the targets, HSP90s, to all of the affected proteins whose modifications changed. And let's zoom in on this cluster here. We have, again, the acetyltransferases and HSP90s and a number of proteins. And if we look closely, and on my poster you can see a lot more detail, there's a number of proteins that affect the cytoskeleton and endocytosis, and there's some cell biology studies that suggest that transfer of early to late endosomes is affected by geldenomycin. That in turn is the effect to affect certain receptor tyrosine kinases. So if we look at individual shortest paths, so from ALK to histones, EGFR to histones, we often see proteins that come up over and over again in these shortest paths. So these have a high betweenness, and I've graphed in this graph betweenness as node size and degree as color. So here's another example. This is the shortest paths between all the receptor tyrosine kinases and two transcription factors that are known to be affected in lung cancer. So what about these hubs? Are they going to be good targets or not? What is the source of this betweenness? The number of modifications correlates somewhat weakly with the betweenness. So these two proteins are the RNA binding proteins NAC and SRRM2. They're highly modified. They have hundreds of modifications. So the number of modifications plays some role. It might make some sense. Modifications, proteins that are highly modified are likely to be signal integrating devices. We asked there is a bias in the protein-protein interaction networks. Many proteins that are well studied, like APP or EGFR, have a high betweenness just because they're well studied. Or they could be sticky. They come down a lot in these high throughput screens. So examples of those are the 14-3-3 class of phosphoserine binding proteins. This is the beta form. This is the zeta form. So the zeta form has a high cluster filtered network betweenness, but the beta form does not. So this suggests that our cluster filtering does filter out some of the bias that's present in the protein-protein interaction networks. So we have hubs, and here's a list of them scrolling by. Highly modified proteins may act as hubs, but that's not the only thing that defines hubs. And these include heat shock proteins, 14-3 class proteins, RNA binding proteins, and adapter proteins that have particular domains. Another look at the data is to normalize against the all PPI betweenness and normalize against the modifications just by putting those in the denominator. And then we get a different look at hubs These hubs that come out are a couple of calcium binding proteins, a couple of adapter proteins. So these are the highly modified hubs. So it's another potential target to examine signaling pathways. So we have RTK signaling pathways that are specific. They have distinct responses. They also have collaborative groups that have common effectors. We have different PTMs, especially phosphorylation and acetylation, that may play dueling roles for signal transduction proteins. Galdenomycin affects this dueling phosphorylation and acetylation, and it affects endocytosis and the cytoskeleton. And finally, we have signaling pathways that converge on network hubs. Some of them may or may not make good drug targets. So I'll stop. Thank you.
Question, so somewhat technical. You were showing these networks with different kinds of edges relating the proteins, presumably different types of interactions between them. When you define things like distances and path lengths and so on, do you deal with the heterogeneous types of edges explicitly, or is there a standard way to do that, or?
I use the edge weights that are, I scale them. So I use just the ones that indicate protein-protein interactions, not text mining. And I filtered out genetic interactions. And I used the weights, but scale the weights to define when I was looking up betweenness. Does that answer your question?
But a path could be 3 different edges of 3 different types. And is that?
Yeah, we have a multigraph.
Right.
There's 2 ways to deal with that. One is to merge the edges, merging the weights, and then you have a single edge, or you can just employ the multigraph. It turns out it doesn't really matter for the path. Okay. I've tried it both ways.
All right,
yeah.
Thanks. Let's thank the speaker once again, and the next speaker come back. So the next talk is work by Alon Diamond and Tamir Tul.